Astronomers estimate that the diameter of the observable universe exceeds 90 billion light years. That's more than 500 billion trillion miles from one edge of the cosmos to the other. The universe contains at least 170 billion galaxies, with stars and planets so numerous they outnumber the grains of sand on all the beaches of the world. Nestled within this cosmic ocean, a sphere of water and rock is home to more than 8 million species of living organisms. This reality has inspired endless speculation, countless theories, and one very intriguing question. What were the odds that life once emerged on the third planet from a sun orbiting within a spiral arm of the Milky Way galaxy? Were we just incredibly lucky? Or was there more involved than blind chance and random interactions of matter and energy? As the human inhabitants of Earth, it is easy to take life for granted. In fact, we're spoiled by its abundance. Our climate and geography support a biosphere that may be unique in the universe. And there's almost no location that's too hot, too cold, or too isolated for living organisms to flourish. Amid this spectacular diversity, perhaps the greatest challenge ever faced by science echoes from every insect, redwood, and whale. How did the first life arise at a moment in time when there was no life of any kind? How did life on Earth begin?
Where do we start? I mean, there are dozens of theories. And you find you've got this wild diversity of viewpoints, many of which are mutually contradictory. To even begin to try to crack the mystery, you have to supply assumptions about what must have happened in the distant past. There's no direct evidence because no one was there to witness the event, and there's virtually no fossil record. What we never observe, ever, is non-living chemicals forming a cell. So in a sense, we have a field of research where the important action has already happened.
The primordial history of Earth is indeed an incomplete narrative. Yet despite an absence of physical evidence, most scientists believe that life started when energy sparked non-living matter in the planet's oceans, crust, and atmosphere to create building blocks for the first self-replicating cell.
When you come to the origin of life, the rules, and this isn't the science itself, this is the underlying philosophy, the rules say to solve the problem, you can use matter and energy and natural law, natural regularities, and chance processes, but that exhausts your toolkit. What you're not allowed to use, fundamentally by the rules, so-called rules of science, is mind or intelligence. If you had to attach a name to this position, you can't do better than scientific materialism, a philosophy that tells you the only acceptable explanation has to be rendered in terms of matter and energy. And if you can't solve the problem using those tools, you're not allowed to change the rules. So from that perspective, how did life come to be via matter and energy alone? Now, try to solve the problem.
All life that has ever existed on Earth is composed of cells, and clues to their origin are woven throughout a network of molecular machines. Enclosed within a modern eukaryotic cell, complex biological mechanisms carry out their work while suspended in liquid. Each of these machines is lifeless, but together they empower growth, movement, metabolism, and reproduction, the vital functions of life. Every component in the cellular factory is made of large compound molecules, and the most numerous are proteins. Functionally diverse, proteins are engineered to unwind and copy genetic information in a strand of DNA, trigger chemical reactions with their hand and glove shapes, control the passage of electrolytes and nutrients through the cell membrane, and tow containers filled with cargo over scaffolds, also made of proteins, that auto-assemble and extend to wherever they are needed in the cell. Magnified 500,000 times, a red blood cell contains 280 million hemoglobin proteins, each designed to carry oxygen. By isolating one of them, we discover that every protein is built from hundreds of smaller molecules called amino acids. 20 different types of amino acids are found in living organisms. The distinct chemical structure of each molecule allows the formation of more than 100,000 different types of proteins. Construction begins when select amino acids are organized into a chain. It's a process often compared to arranging alphabet letters into meaningful words and sentences. If these individual building blocks are sequenced correctly, the chain folds into a protein fully equipped to perform its task within the cell. But if the amino acids are arranged in the wrong order, the chain won't fold and is eventually destroyed. The simplest living cell contains at least 300 different types of proteins and the biological machinery necessary to grow, reproduce, convert energy, store and process genetic information, and protect its contents from the outside world. Similar components and functions were also required in the first living cell. Now let's apply this basic chemistry to a familiar theory for the origin of life. Scientists have long speculated about primordial oceans loaded with amino acids that floated around like bumper cars. Perhaps some of these random collisions produced a chain long enough to fold into a protein. Later, more amino acids linked into more chains that folded into even more proteins. Simultaneously, other complex molecules, including nucleic acids, fats, and sugars, also assemble themselves into essential components. Then, without foresight or direction, just the right combination of molecules somehow came together in this chemical soup to form a membrane that encased all of the specific machines required for a self-replicating cell. And there you have it, life from non-living matter.
You know, it all sounds so reasonable. The problem is, on the early Earth, the internal complexity required to survive it all would be overwhelming, because life as we actually know it is characterized by molecular machines and lots of them. And if you take away that essential hardware, the cell ceases to be. Because there comes a point when you can't get any simpler and still exist. Below that means death. One sometimes hears the criticism, you can't take modern cell biology and project it into the past. Because whatever the first cell was like, it was a lot simpler than what we see today. All right, I can entertain that as a premise. Describe the first cell.
If you're going to be the first living cell, you need a boundary, a membrane that protects you from the rest of the environment. This membrane must selectively allow the passage of specific nutrients and raw materials required for fuel and energy. You'll also store huge quantities of genetic information in coiled strands of DNA. These detailed instructions for life are copied with remarkable precision. Then in factories called ribosomes, this information is converted into chains of amino acids and proteins. Finally, you'll be equipped with everything you need to reproduce future generations of cells.
These basic functions are performed by all living things. They're, in a sense, diagnostic of what it means to be alive. So whether you were a cell in the past, a very simple cell, or a more complicated cell today, really doesn't matter. You're going to have to perform these functions.
Despite daunting obstacles, the notion that basic chemicals once arranged themselves into a living cell has been suggested for at least 150 years. In 1871, Charles Darwin wrote a letter where he described a warm little pond filled with all the right chemicals to create life. Half a century later, the Russian biochemist Alexander Oparin expanded Darwin's ideas. In his book, The Origin of Life, he theorized a primordial soup and a gradual progression from simple chemistry to a living cell. Then in 1952, this hypothesis was put to the test. Stanley Miller, a graduate student at the University of Chicago, set out to determine if simple chemistry could produce actual building blocks of life. The young scientist constructed a glass apparatus to simulate the ancient water cycle and filled it with a mixture of gases that were thought to comprise the atmosphere of the primitive Earth. Miller heated the components, then charged the gases with bolts of lightning, electric sparks to trigger a reaction. Within days, a tar-like sludge formed in the glassware. In it, Miller identified 4 of the 20 amino acids found in proteins. For the first time, under presumed natural conditions, molecules crucial to life were generated from matter and energy.
There was tremendous excitement when the Miller experiment was first published because it looked as if it was easy to get the building blocks of living things. And if it was easy to get the building blocks, maybe it wasn't so hard to get from those chemicals to the first living cells.
While groundbreaking in its premise, Stanley Miller's experiment later proved to be deeply flawed because he hadn't used the correct gases, neglecting nitrogen, carbon dioxide, and oxygen that would have destroyed molecules required for life on the early Earth. But despite these errors, Miller's work inspired more attempts to recreate prebiotic conditions. Today, after decades of investigation, hope for a swift, clear pathway to a solution has long faded. Yet origin of life research continues throughout the world. Its core hypothesis, that living cells emerged from natural chemistry, is now known as chemical evolution.
Chemical evolution comes with a lot of baggage. And if you're going to take it seriously as an explanation for the origin of life, then you also need to take seriously certain fundamental properties of biology.
The molecules involved in life, they have no intelligence, they have no foresight, they have no way of knowing what they need to do next in order to get themselves assembled into organic chemistry. If I put amino acids in a test tube in my lab, even if I added heat and shook it up real well and kept doing that for 100 years, or 1,000 years, or 10,000 years, or 1,000,000 years, nothing would happen.
Amino acids, nucleotides, lipids, by themselves, they do not represent life. They're inert. They're not reproducing, they're not storing information.